Good morning, ladies and gentlemen, and thank you, Janet. It's a pleasure for me on behalf of the Ted Rogers Center of Heart Research to chair a few of the speakers that I'm so looking forward to hearing from this morning. After 7 really inspirational laureate presentations yesterday and the ceremonies last night, this is just the icing on the cake with another great menu scheduled. As Janet mentioned, the Ted Rogers Center is a relatively new entity. It was delighted to help sponsor this morning's symposium because we really are at the intersection of genomics, bioengineering, regenerative medicine, and clinical medicine in our mission at the center. And I think the first 4 speakers that I have the privilege of introducing this morning sort of reflect both that breadth and ingenuity. So our first speaker is a local talent up the street. Emma Allen-Vercoe is a professor at the University of Guelph who is a microbial ecologist. I very much enjoyed her TED Talk where she coined the phrase I had not heard before, the poo signature. She will, I think, tell us that our biology and in fact also our genetics are more microbial perhaps than human. She's a highly regarded teacher, a very well-funded scientist, and importantly, she was the lead author of the Canadian Working Group report on fecal microbial therapy. Emma, welcome.
Thank you very much. It's an absolute honor and a pleasure to be here today. And thank you very much to Janet for inviting me and to the Gairdner Foundation for supporting this very important venue. I think it's an extremely exciting and inspiring event. And I'm really, really happy to take part. So I hope I can do microbial ecology justice in today's talk. So I'm going to try to tell you a little bit about why your microbes, particularly the microbes in your gut, are so important. And so hopefully you're not going to be too grossed out by some of the things that I'm going to tell you about, because necessarily a lot of what I talk about is poop. But it's not actually poop metaphorically, it's real poop. And I think it actually contains a lot of extremely exciting opportunities for the future. So the first thing I need to tell you is that you're actually not human. So that might come as a surprise to some of you. You are superorganisms of human and microbial cells. So we should stop thinking of ourselves as human and start thinking of ourselves as these superorganisms, and that makes us sound really cool, that we're superorganisms, I think, so that's a good thing. But we should really start to appreciate that the microbial side of us, even though we can't really see it because microbes are about 100 to 1,000 times smaller than animal cells, so we don't really see these microbes, but they play an enormous role in our health, and that's very important to get across. Now the thing is that we exist in an equilibrium with these microbes, and this equilibrium we've come to realize in the last few years or so of research in the microbiome field, which is really accelerating at a massive rate. We've come to realize just how important this equilibrium is and how easily it can be disturbed, and that's kind of at the heart of what we think is many chronic diseases today. So the big question here is, how human are we? Now, you may have heard some reports that you're only 10% human and 90% microbial. Actually, so I'm here maybe in part today to help bust that myth a little bit, because it's turned out that that was based on some rather shaky math. So a little while ago, a group at the Weizmann Institute in Israel actually set out to really try to calculate this number more accurately. And what they found, using a reference human of 70 kilograms, around 20 to 30 years old, and 1.7 meters tall, they figured out that there were about 30 trillion human cells within that individual. And then if you look at the microbiome, that's the collection of microbes that live on them and in them, that would represent about 39 trillion bacterial cells. Most of those actually live in the gut. So that actually means that, in fact, we are only 1.3 ratio of 1.3 to 1 bacteria to human. So we're not quite as microbial as we used to think. But it is important to understand that they're still in the majority, and that it's not just the number of cells that are important, but in fact the amounts of genes that these microbes actually carry. So there's about 100 times more microbial genes in your microbiome than there are human genes in your body, and so that's quite an extraordinary number if you really think about it. Now, most of the microbes, of these microbes that live on and in your body, actually live in your gut. And the gut microbiome, as it's called, contains many different microbes. I've focused on a number over the years. Today I'm really just going to be talking about the bacteria, but I would be remiss if I wasn't pointing out that there are other things in there as well. There are viruses in there. In fact, I work with a group in San Diego looking at what we call the human microbiome, which are the normal viruses that live in your gut. And mostly in your gut. And we also look at bacteria, obviously. Yeasts are becoming increasingly important. There's archaea. These are kind of ancient bacteria. They're not really bacteria. They're about as related to bacteria as we are. But they carry out some really incredible metabolisms that are very important because of their ancient heritage. They've had a long time to evolve for these amazing metabolisms, so we shouldn't forget those either. And then there's also protists. Now, these are the eukaryotic microbes, the little critters. We know very little about them, but they're there. And so all of these collectively make up your gut microbiome. Now, each gram of feces contains about 10 to the 11 bacterial cells. That's about 10 trillion cells in the average bowel movement. So after you've had a bowel movement, then very briefly you become more human, which is quite funny if you really think about it. If you feel more human after a bowel movement, that's probably because you are more human after a bowel movement. Now, everybody's gut composition is different. So this is work that was done as part of the Human Microbiome Project a number of years ago, which I was a part of. And I'm just focusing now on the stool component of this. So what happened in the Human Microbiome Project in the first stage of it, that started in around 2006, is that we went out and found 139 really healthy individuals. They were screened for their health. They were mostly in the Houston area and Washington University St. Louis area, just necessarily because that was where a lot of the research was being done. But these 139 volunteers were poked and prodded. Samples were taken from every orifice of their body, practically, and then sequenced to see actually what was there. And what we found was that in these 139 individuals, you can see in this bar plot here, so if I can pull the cursor up here. So this bar plot here, every vertical line here represents one of the individuals from this cohort. And hopefully you can tell that when we stack them all together like this, that everyone has a very different microbial composition. And this is actually at the phylum level, which is a very high taxonomic level. So if we can see differences here, then those differences will expand as we go down each taxonomic level. So you can see that there's an awful lot of difference between all of those individuals. And so in fact, that's where the term poo print comes from, or the poo signature, because everyone has this unique microbial signature that's unique to them, and that actually pretty much stays with you for most of your life, from the time that you're weaned to the time that you reach old age, unless you mess with it, and I'll tell you more about that in a little moment. But what was really incredible is when we sequenced everything in there, so we looked at the metagenome, what we found was the fact that all of these functions, so when we bin all these functions together, the functions are very similar. So it's really not so much about what microbes are present in someone's gut, but it's about what those microbes are doing that's important. So what are they doing? So what do these microbes do for us? Well, they do a lot of things for us, and we're just beginning. We're really on the cusp of understanding how important these microbes to us. They're like little pharmaceutical companies in our bodies working to make a lot of molecules and carry out a lot of functions to help us survive, to help us live. We now recognize that they regulate the immune system. They help to actually educate the immune system in many ways, and that's a very important part, especially during early life. They help to extract energy from food. They control potential pathogens, and we used to think that was just through competitive exclusion, but now we actually realize that they actually have interactions, direct interactions with some pathogens that actually tell these pathogens to turn off their virulence determinants and behave non-pathogenically, which is a very exciting new area, and I'm not talking about today, unfortunately. And these microbes in our guts in particular make some essential metabolites, and they include things like vitamins and cofactors. Vitamin K and vitamin B are some important vitamins that are made predominantly in your colon. The microbes in your gut improve the intestinal function. They actually help your intestine to function properly through interactions that are still rather mysterious. There's potentially some interactions between neurons in the gut. So there's the nervous system in the gut that is detecting what's going on in the gut may in fact be fed in part through signals from the gut microbiota. And then the microbes there also remove toxins and carcinogens, so that's a very important role that they play, probably rather underappreciated. And so altogether, all of these things, they make you really think that your microbes in your gut can actually be thought of as a virtual organ. And really, collectively, they do as much work for you as the average liver. And no one in this room would believe that you could do without your liver, right? So I think it's really important to understand that, that you shouldn't think that you can do without your gut microbiota either. Now, a little bit of Ecology 101. So ecosystem biodiversity is actually what drives overall health of an ecosystem. And what do I mean by that? So I just mean that if you have a high diversity of species, and think about a rainforest that has a huge diversity of species, that rainforest ecosystem is considered to be a healthy ecosystem because there's balance within the ecosystem. There's plenty of species, they all have different functions, but there is functional redundancy. And what do I mean by that? I just mean that if some species within that rainforest were to go quietly extinct, and that species, within that huge repertoire of other species, there was another species that could step up to do the work that now extinct species could do, then from an outside point of view, you probably wouldn't notice the extinction of that one species, that first one species. And when you have this sort of situation of high gene count, essentially, that helps to build in this functional redundancy, which actually turns out to be extremely important to the homeostasis of the ecosystem. And it provides, for example, things like resistance to damage, so that when damage happens, then the ecosystem can recover nicely because there are other species that can step up to take the place of the damaged ecosystem whilst the damage is healed or rectified in some way. Now, if you contrast that to an ecosystem where you have a low diversity of species, a sick ecosystem, if you will, there you have imbalance. You have a problem because you have functional disability. And when you have functional disability, it means that if something was to go extinct, then the whole ecosystem may suffer for that because there is nothing that can take the place of the function that's now lost. And so the ecosystem collapses, and that's a rather sad situation. It's very difficult to bring it back to health from that situation as well. And it also means that that ecosystem is inherently much more susceptible to damage. Now, this is, I've given some analogies of a rainforest and a kind of sick-looking forest there, and that's at the scale of the macro, the animals, the plants, but it's also true at the microbial scale. These rules still apply. So the question here is, are we damaging our health by eroding microbiome diversity? So there's a few hypotheses, some of which you may have heard of, and a couple which you may not have done. The hygiene hypothesis, I'm sure everyone has heard of. That's from 1989. This states that a lack of exposure to certain infectious agents during the childhood years actually drives allergic disease. And it was a sort of first kind of look at maybe what's going on in terms of infectious disease, and could it be related to the fact that we live a very clean lifestyle now. What's interesting is that didn't really, back in 1989, didn't really look at the microbiome at all. In 1989, we just didn't really appreciate the microbiome at all. We used to think that, definitely we knew that there were microbes in the gut, for example, but we thought that they were just hanging out there, being passengers, if you will, not really doing too much, not really helping us in any way, and just hanging out. And we couldn't have been more wrong. Now, in 2003, Rook came up with the old friends hypothesis, which is a little bit more stepping in the microbiome direction. And here he was suggesting that humans are dependent on a co-evolved microbiome to educate the immune system and prevent inflammatory disease. And so here's the first kind of indication that the microbiome might actually be important in shaping the way that our bodies kind of develop. And that may have come from the fact that we have co-evolved with these microbes for millennia. That's why they're our old friends. And then finally, the missing microbiota hypothesis, I think, is probably the scariest one of all. Now, this one states that a loss of microbiota generally compounds over generations, and recent changes in lifestyle have greatly exacerbated this loss. So what does this mean? It means that the thought is that because you end up getting a lot of your microbes during your very early years through the process of birth and your immediate environment, and then your microbiome becomes set about the time that you're 3 years old for reasons that we don't quite understand yet, that very early environment is extremely key to the eventual health of an individual. And so if each successive generation is clean and hygienic and microbes have been lost from that system, then each successive generation will have a compoundedly less diverse ecosystem, and that's a problem that we see. And some of the things in the animations there you can see that might be driving this are the fact that in the Western world we live an increasingly sedentary lifestyle, we eat a terrible diet, and we take a lot of drugs, of which antibiotics being the obvious ones, although antibiotics are life-saving drugs. I'm not against antibiotics, but they have not been used particularly well, especially in the last century. I suppose is in the 80s and 90s is probably the height of their bad use. We're kind of paying for that now. And so this is a really scary hypothesis, and probably what we're seeing in terms of what the authors were suggesting is that what we're seeing in terms of the rise of chronic disease could actually be related to the lack of microbial diversity that we've inherited from our parents and previous generations. So examples of diseases that are associated with an altered gut microbiota diversity, and this is published research, you don't have to look very far now. Microbiome, as if you know it, it's a very attractive subject, a very sexy subject in the literature right now. There's an awful lot of literature that's being written on it. And there's an awful lot of diseases that keep popping up where the researchers are seeing that there are changes in the microbiome of people that suffer with these diseases. Now the problem is that this kind of creates a situation where people think that the microbiome is going to cure everyone. And I would caution that we need to know far more about it, because the reason for that is that association is not the same as cause. And it's very easy to find an association. Finding a cause is actually extremely difficult to do. And so we're getting closer with some of these diseases in particular in seeing the connection between cause and the diseases that some of them there. But the one that I really want to point out today, that I'm kind of going to take the rest of this talk in this direction, is Clostridioides difficile infection, or it used to be called Clostridium difficile infection, or C. diff. That one we actually know is a disease which is caused by an altered gut microbiota diversity. Now the pervasive advertising message is that all microbes are bad. And so you just have to look around you to see how many things you can buy off the shelf in the supermarket that are antimicrobial, and all the advertising that we're faced with that suggests that we need to live in a really clean environment. And I have a real problem with that. I think that we need to really start changing the way that we're thinking about our environments. And having a clean environment doesn't necessarily equate to having a sterile environment. Only a tiny, tiny fraction of microbes are actually pathogens. And now some of those pathogens are very important pathogens. I don't want to downplay the importance of them. But as well as sort of understanding that there's only a very small minority of them, what microbiology has done a big disservice to in the past decades is understanding about the normal microbes. We have failed to study them. It is not very sexy to study health and to understand what health is, what health research is. And so we've tended in microbiology to study the pathogens. And when you study the pathogens out of the context of the normal microbiota, you don't necessarily see the whole picture. Now, antimicrobial practices don't actually discriminate between pathogens and microbiota. So every time you use an antimicrobial substance, then you're actually doing damage to your microbiota because there's collateral damage. And that includes antibiotics. Again, I'm not demonizing antibiotics because antibiotics are very important in medicine, and you should take an antibiotic course if you're prescribed one. But we have to remember that when we take that antibiotic course, we are doing damage to the microbes in our bodies as well. Collateral damage, sometimes necessary, but we have to appreciate that happens. Now, some microbes are like bad teenagers in a subway station, okay? So some microbes do not, we call them opportunistic pathogens, okay? So in a crowded environment, they tend to behave well. So just imagine a subway station full of people, and you have a few bad teenagers there. I have a teenager, so I have an authority on bad teenagers. In a crowded environment, these teenagers might actually behave fairly well. They may actually be upstanding members of the community. They may give up their seat for an elderly person. They may pick up something that someone has lost and return it to them. And that's a sort of a social norm that we might see. Now, the problem arises when the train comes by, picks all the people up, and leaves those bad teenagers alone on the station. Now they have no one to police them, or no one to oversee what they're doing, no peer pressure, if you like. And when that happens, they can start pulling out their weapons, if you like, their spray cans, and doing damage to the subway walls. and a little bit like C. difficile, I'm going to tell you in a minute, can start doing damage to the gut wall by pulling out its weapon of choice, which happens to be a group of toxins. So the example bad teenager is Clostridioides difficile, or C. diff. So in C. diff, you have a normal colon usually with an abundance of microbial diversity. Everything seems to be fine. You might have C. difficile. A lot of people actually are colonized by C. difficile, but it's there in very low numbers if it's there at all. What happens is that you take an antibiotic, and a broad spectrum antibiotics such as clindamycin, cephalosporins, or ampicillin are usually involved in this disease. And that antibiotic might be for nothing at all to do with the gut. It might be for a completely different infection, perhaps a dental infection, something like that. But what will happen is that because of this collateral damage I was telling you about, you'll have a sudden reduction in microbial diversity. Your C. difficile starts growing to high numbers. And when it grows to high numbers, there's an image of it just here, so you can see what it actually looks like. This is a vegetative cell, and here is a spore. So this is an endospore. This is what this microbe does when it perceives a difficult situation. It sporulates, and when it sporulates, those spores are very, very difficult to kill because they're like in suspended animation. Antimicrobials will only work on cells which are actually metabolizing effectively. And so C. difficile actually got its name because it's very difficult to work on because it sporulates at the drop of a hat. So it's actually very, very difficult to keep it in an environment where it doesn't sporulate and where you can actually study it. So you have a situation where C. diff is growing into high numbers, so it'll be in this form, what we call the vegetative state here, even though that seems like a weird term. That's the state that we use for microbes which are growing actively. So it grows to high numbers now because there's plenty of room for it to do that because the rest of the microbes are now missing. They've been killed and they're removed. And so there's space within that ecosystem for these C. difficile to expand, rather like dandelions will expand on a lawn which has been raised to damage. The dandelions will take advantage of that situation very quickly. C. diff will take advantage of this situation very quickly. When it grows to high numbers, it signals to other microbes, or to other of its own species, to understand that it's there in enough numbers that it can start causing some damage. And it actually creates or produces a series of toxins. We call them C. difficile TCDA and TCDB. And there's a few others as well that are coming to the front. But what's important to know about these toxins is that they damage the gut wall. They first of all lead to diarrhea, and it's a particularly purulent form of a very unpleasant diarrhea. And this diarrhea, if it carries on, you'll end up with ulceration of the colon. There's a really nasty picture of what we call pseudomembranous colitis here, which is a hallmark of this disease. This is from someone's colon, an affected person's colon. It looks very painful, as you can see. And if that ulceration of the colon isn't corrected, then that could actually lead to death, and particularly in immunocompromised elderly people. So what will usually happen is you'll go and see your doctor well before the signs will get really too critical. And the doctor will usually give you an antibiotic to try to get rid of the C. difficile infection. And those two antibiotics that are generally used are vancomycin or metronidazole. And then what will happen is that those will kill the C. difficile, the vegetative C. difficile, and the symptoms will abate. And then you'll stop taking the antibiotic therapy, and the hope is that you'll return to normal, that your microbiota will have recovered enough that the C. difficile can once again be kept at bay. But what we're seeing is relapses in around 10 to 33% of individuals, and so these relapses happen, and the patient has to go back on antibiotics. And we call this a revolving door situation where these patients are constantly either on antibiotics or in the clinic seeking treatment for C. difficile symptoms. And it's a real problem because it's extremely difficult to treat. But recently, there's been a little bit of headway being made because fecal transplants have been shown to actually be useful and as a therapy for this disease. So fecal transplants, here's the gross bit, so I warn you now. Fecal transplants are exactly what they sound like. You take someone's poop sample, you grind it up in some saline, and you introduce it into a patient. And you can introduce it into a patient in a number of ways. You can do that through a rectal enema, which is the simplest way to do it, but that doesn't get very far up into the colon. You can do it by colonoscopy, which is actually the preferred method, I think, in Canada, but obviously requires clinic time to do it. You can actually, in Europe, it's given through a nasoduodenal tube, which really doesn't bear thinking about too much. And in Calgary, my colleague Tom Louie has been experimenting with putting freeze-dried or frozen stool into gelatin capsules and having patients swallow these capsules. And you'd be amazed how many patients sign up for it, because this really is a very serious and unpleasant disease, and a lot of patients are very desperate. Now, the amazing thing is that it actually works, okay? So it results in cure of the patient in over 90% of cases. And there was a clinical study, a clinical trial that was done looking at fecal therapy, fecal microbial therapy as it's called, and comparing it to vancomycin as clinical trials do as the control. And that trial was actually halted because it was deemed that the vancomycin, those patients on vancomycin, were actually ethically less at a disadvantage, because they were far less likely to benefit. And so it was the fecal transplant was working so well that they stopped the trial and gave everyone a fecal transplant. This is very rare in medicine to have a success rate this high. So there's really something very good going on. There's rapid resolution of disease and only rare recurrence of the disease as well. But, now there's always a but. So we don't yet understand the potential negative health implications of FMT. How do we screen for things we don't know about? If all the donors are different, which is often the case, this has very little scientific value. It might be valuable for the patient, but we actually, as scientists, don't learn very much from what's going on, because I told you everyone has a different gut microbiome. And that also changes slightly between donations for a single individual, depending on the diet that the donor is eating as well. We can actually get over that to a certain extent by using what we call super donors. So these are donors that volunteer their services, if you like, every morning to provide a stool sample that's used in the hospital clinic. And these donors also consent to having multiple checks of their health and very regular times, so that we can make sure that they're screened constantly for the absence of major diseases that can be passed through stool. But the problem with these super donors is that super donors can get sick as well, and so that's also a problem. And for C. difficile infection, we know that the benefits are really clear, but the long-term effects of fecal transplant altogether are really unknown. We just haven't really got far enough in science to understand whether what might be going on might actually have some long-term consequences for the patient. Here's an example in the UK, the Taymant Clinic, which is actually not a real medical clinic, but it's a naturopath clinic that's set up to look like a medical clinic, is actually providing fecal transplants as a service to the National Health Service there for C. difficile infection. But as well as that, they've started branching out into treatment of just about anything you can think of in response to just all you need to do is find a donor, and they will screen that donor for you. And then the funny part that I think, anyway, it's funny, it's kind of dark humor, is really that they, to make sure that this poop sample is safe to give to a donor, they basically put it in the freezer for 3 months. And then they figure that it's safe because the donor hasn't actually got sick with anything. But I've told you that everyone has a different gut microbiome. That includes a different set of opportunistic pathogens. And some of those opportunistic pathogens may actually activate in a different recipient. And so there is a risk as well. I don't think a 3-month freezer quarantine is going to really help with that. And this is just one example, anecdotal evidence in the literature that shows that sometimes things can go wrong, fecal transplants. So this is a case of a woman who was normal weight before she got C. difficile infection. It was C. difficile was very difficult to treat, so it was decided to give her a fecal transplant. She was given a fecal transplant from her daughter, who was overweight, and this poor woman started putting on weight. She got better from the C. difficile, but she started putting on a lot of weight. Now, it's an N of 1. We can't really draw too much conclusion from that, but it really makes us think we should really start to be a little bit more careful. So we need to start rethinking this therapy. So instead of stool, why don't we use pure microbes that are isolated from the gut of a healthy person? There are several perceived barriers to that. The first one is that most of the gut microbial species are unculturable. This, again, is perceived. That live microbial therapeutics are hard to standardize and thus regulate. And then how do we know which microbial species to include? So the first one is really not true. So there's about 100 to 200 different bacterial species within the average person's gut microbiota. This species richness has previously been drastically overestimated again, that now we're down to about 100 to 200. The major barriers to cultivation are low abundance and fastidiousness that can be overcome through the use of bioreactors, sorting techniques in particular culture conditions, and also patience, know-how, and hard work. And that's kind of what we do in my lab, is we use bioreactors to actually grow the microbes from the human gut. And we do that because we recognize that the human colon is actually a very sophisticated bioreactor, and chemostat bioreactors can be used to approximate the human colonic environment. We can modify and control pH, temperature, food that we feed it, the flow rate, and the oxygen concentration. And as a result, we've created what we in my lab call the RoboGut. And we have, thanks to CFI and to the Ontario Ministry of Research and Innovation, we have 2 sets of these now, so 12 vessels that we can actually use to mimic at the same time 12 different people's colons if we wanted to. And these can be used to help us culture these so-called unculturable microbes, because it turns out that these microbes are culturable. We just need to know the conditions into which we grow them, so we emulate what happens in nature. So using pure gut microbes or pure gut-derived microbes as therapy. So this represents our first attempt, and I'm going to whisk through it because we have published this. We made something that we called Repopulate, and we published this in 2013. It was a mixture of 33 bacterial strains. And we've since named it MET1, just because journals don't seem to like the name Repopulate, don't know why. So we have 33 bacterial strains from one healthy donor. And we basically purified these microbes, made a slurry, and it looked like a sort of milkshake in a suspension in saline, and we gave 100 ml dose via colonoscopy to 2 cases of severe recurrent CDI infection, and both patients recovered within 48 hours and remained C. diff free ever since. And then this was really the genesis of the company NuBiyota, which we founded in 2013 to carry this work on a bit further. So you might be asking, why on earth did we stop after 2 patients? And that was really because Health Canada were very concerned that when we were doing that trial, they had given us permission to do that trial as a probiotic trial, which is regulated under the Food Directorate. But however, this is very different from a probiotic. These are human-derived microbes, and so they asked us to reconsider the trial and to reclassify what we were producing as a first-in-class biologic drug, which is about the other end of the scale, the hardest type of drug to get through regulatory profiles. So these things are very hard to regulate. They are very hard to standardize. But a defined mixture of microbes should be easier to standardize and to regulate than actual feces. And regulation is necessarily difficult, but really someone needs to take the first step. So that's really what we've been doing with NuBiyota. And we've been working to develop a new framework for standardization to define a first-in-class biologic drug, and we call this microbial ecosystem therapeutic, or MET. So the third consideration is which microbial species should be chosen. So we need to think about the needs of the ecosystem. And for that, that's really where I come in as the microbial ecologist in the team. We're very interested there in stability, diversity, functionality, and strain interactivity, so how these microbes actually work together to form a medicine which can be synergistic. And then on the other side of the coin, my investors are very interested in things such as cost and safety, of course, culturability, reproducibility, and integrative ability. But we also have to consider the patient, the needs of the patient. And I think everyone here would appreciate that most patients would much prefer to take an oral capsule than they would to put something up their rear end. And so that's really, as well, the other part that we've been focusing on. And so the enhanced safety is very good. Fecal transplants can't be undone. Once you've given someone a fecal transplant, there's no way of getting rid of it. But microbial species used for MET have been screened and confirmed generally sensitive to selected antibiotics, and so there is the potential to remove them if something was to go wrong. The genomes are sequenced. They're checked extensively for the absence of known virulence or resistance genes. And because they're well characterized, we can start to track each strain during and after treatment of a patient to really start to understand what actually might be going on. So in other words, the science benefits as well. So where are we at? So MET1, if you remember, I said it was trialed as a novel probiotic. It's a pure culture, has a suspension of microbes for delivery via colonoscope, and there were 33 bacterial strains of 25 bacterial species. And in the last 5 years, what we've been able to do is develop a first-in-class biologic drug and get it through the CTA process. This is a pure culture as a stabilized, lyophilized capsule form for oral delivery. So very much like how you would take a probiotic, and we have included greater diversity because we've learned a lot in the last 5 years. The phase 1A trial, I'm very happy to report, is currently underway in Kingston, Ontario. And this is for the treatment of recurrent C. difficile infection that's repeatedly failed to be resolved using antibiotic therapy. So just the last couple of slides, moving towards the future, how are we going to approach the development of novel mets? Now we've done this, we're blazing the trail for this, how can we improve this product further? So what you need to do is you need to consider the gut microbiome as the engine of your body, right? The metabolic engine of your body, if you like. And so when you've got an engine that's not running right, then you can really see that it's not running right by looking at the emissions that come out of it, right? And so we need to start looking at what's wrong, can we replace the faulty parts in order to get the engine running optimally again. And this is very much going towards the idea of personalized medicine. How can we screen to see what might be wrong with someone's microbiome to put it back together? And the answer is that we need to look at the metagenome, which is really a blueprint for all the biochemical processes that go on within an ecosystem. And we need to look at what we call the metabolome, or the metametabolome, which is all of the little molecules that are made by these microbes as a process, as a part of their metabolism, that we can actually measure and understand what they're doing. And if we look at these two things, then we can see what might be wrong when we can start to replace missing functions to get the engine running optimally again. So with that, I just want to acknowledge the support of all the wonderful people that have funded me over the years. Things are really starting to take off now, so I'm very excited. I have my academic side of the lab and also the NuBiyota side of the lab. We're all in one space at the moment, but NuBiyota will be moving to new premises soon. They're all wonderful people that work with me. They work extremely hard, and I'd like to take this opportunity to thank them very much for their very hard work, and to thank you for your attention. Thank you.